everyone, a very warm welcome to all of you in lovely professional university Edu-Webinars. My name is Dalvinder Kaur and I'm your host for today's webinar. As I can see, people are still joining, so let's wait for a few more minutes so that everyone can join. Till then, let me take you all on a virtual tour of our beautiful university. So here it is.
Finishes on 90 meters, 23.
Thanks everyone for calling. So let's talk about the call response, especially on option C. Is this video real or AI? No, this video is absolutely real video. No, AI is harmed in making this video. So for the people who wants to see this video again, I'll share the link in the chat box later in 2-3 minutes. So as I can see, everyone has joined. So our today's topic is Biotechnology 2.0, redefining life through innovation. Before we proceed further, allow me to introduce the expert of the day, Dr. Himanshu Singh, Professor, Department of Bioengineering and Biosciences at lovely Professional University. Before we proceed further in today's session, I would like to request everyone for any question. You can write in the chat box or in the Q&A box, and same will be addressed on my side in the end of this webinar. Now I'm going to hand over the session to our expert, Dr. Himanshu Singh, and it would be an absolute delight to listen from him. And I'll be joining you all as the attendee, as it's always great to learn something. Over to you, Dr. Himanshu Singh.
Okay, thank you, ma'am. I'm Dr. Himanshu Singh. I'm the head of department in Biotechnology and Bioinformatics Division in the School of Bioengineering and Biosciences. Now, today we are going to start our journey to see how the biotechnology is transformed. Because you already know about the classical biotechnology, where we are utilizing the genetic engineering, where we are utilizing the concept which are going to be modify the biology in a way so that we can utilize it for our purposes. Okay. Now the era has changed. After the 2022, you can call the launch of first AI model, that is the ChatGPT. Everything is changed. Now the biology is moving in a different direction. I think I want to present something in front of you. Now, please see the Biotechnology 2.0, that is the redefining life through innovation. What we are doing currently, we are entirely changing. We are entirely changing the era of the biotechnology now by the utilization of the AI and by the utilization of the machine learning tools. Now, what the AI and machine learnings are going to change, how it is going to change the biology, how it is going to change the biotechnology. Okay, a new buzzword which is coming nowadays, Till now, we are calling it as a biotechnology. Now the new word is tech bio. Okay. Tech bio means we are utilizing the technology for the benefit of the biology. Now we are required to utilize the technology and utilize the biology into the technology so that the technology is going to be benefited by the application of the biology. This is the entirely different. If you think about it, the concept that five, okay, that apply the biology for the technology so that we can meaningful research by which our area, our lives, our healthcare system is going to change in a big way. Just see here, the foundation and the foundation of this change is basically from movement from the classical observation to the synthetic design. Synthetic design means creating a new genome, creating a new molecule, creating a new organism, so that the all kind of inabilities or the things where the humans are not able to perform, they can perform. Okay, you can think about it as a movie, but not, it is a reality which is going to be happen next 5 to 10 years. And you how the AI and machine learning is going to change the entire biological sector. The biotechnology 2.0 is defined by the application of these technologies for the benefit of the biological sciences, to find some treatment of the disease, to find new ways to create new genomes, to create new kind of organisms. Now, if we are talking about synthetic design, what is the synthetic design? Synthetic design means you already know what is the genetic code, ATGC. In RNA, you are having the uracil also, but ATGC is the basic genetic code. Now, only the 4 codes are creating your whole genome. They are going to create the protein, they are going to create the amino acid, and every function of your life, every function of your body will be dependent onto these 4 only, ATGC, adenine, guanine, and cytosine, as well as thymine. Okay. Now, if we are talking about the biotech 2.0, the foundation is how we are going to decode the natural language. Okay. The genome is a kind of a natural language of the life form. Now, natural language, if you are able to decode the natural language, and in your own way, you can rewrite it and translating into the new biological blueprint, then it is going to create a kind of a modules which are basically modified the humans in a way so that they can achieve which is not possible till now. Now, if you are talking about the basic difference between the biotech 1.0, which is called as the classical era of the biotechnology, and the biotechnology 2.0, which is called as the synthetic era of the biotechnology. Till now, we are harnessing the ability of the cells, the microbial cell, the animal cell, the plant cell, to produce something. Now we are going to create the cells which are required for us to produce something. Okay, it is an entirely different approach. Now, if you see in biotechnology 1.0, discovery and natural selection is a major parameter. Natural selection as per the nature want. Nature want that this cell is the more suitable for the production, we are utilizing that. But now in biotechnology 2.0, what we are going to do? We are entirely changing this concept. Now we are de novo design the new cells according to the wish of, or according to the process requirement. We can engineer the cell. We can change the entire DNA of the cell so that they can perform a new kind of a thing which is not the natural. Okay. Now, core tool set. If you in biotechnology 1.0, basically we are moving from the recombinant DNA. We are utilizing the PCR, the various variants of the PCR, and the basic hybridomas. Okay. Now, in biotechnology 2.0, we are moving towards the gene editing. Gene editing, the classical example of the gene editing, one of the child which is born in US around 2 years before, they have what they have done. They have modified its genome because into the family they are having one of the genetic disease. And now by the application of the CRISPR means basically the gene editing tool. Now we have modified the gene of an individual, and the individual is free from the genetic diseases. I think you have already heard about the genetic diseases do not have any cure. We can manage it, but because it is in our DNA and our DNA cannot change. But now the era is going to come where DNA itself is going to change. You are responsible for change the DNA. You are responsible for removing the diseased part of the gene, and you are going to replace them with the healthy part of the gene. The CRISPR-Cas9 technology, the base editor, the machine learning models to how to design the new organism, how to design the new genome. Now, workflow are basically, initially we are doing the experiment in a triplicate. Why? Because sometimes the science is kind of a thing which is dependent onto the trial and error. Okay, you are going to try something. It may be possible you are not getting your own result which is required. Okay, that is called as the error. Now the manual screening. Always the one of the person is required to be there to check how the reactions are completing. Okay, but now the things are moving towards the automation. Automated design, build, test, learn loops. Okay, you are just required to give your sample, and rest all the things will be done by the automated robots. Okay, these are basically the small robots which are going to perform the different kind of a function. Different kind of a function included your scientific experiment also. Okay, till now, because of the different handling of the individuals, sometimes we are not having the result which are very great and which are required from the process. But now, because of the less human intervention, the automation is going to create a era where the treatment of the diseases which were not available till now. Just take an example. Around 6 months before, I have heard about one of the news from the Stanford University, that in Stanford University, scientists have developed one of the method for the treatment of the one kind of specific cancer. Okay, the cancer, the research is going on from the last 40 years. The continuous research, billions of dollars are already invested into the cancer research. But till now, we are not able to get any foolproof disease treatment for the cancer. Now, in Stanford University, the scientists are telling we have developed one of the method for the very specific kind of a cancer treatment. This is just a initiation or nascent stage in your AI application into the biotechnology. Okay, so first of all, and afterwards you can see what is going to be happen. The entire healthcare sector is going to change. Now, if you remember if you study during the +2 also, how the drugs are going to be made. Okay, drugs are basically require 15 to 16 years from the development to come to for the human consumption, because the clinical trial is very lengthy. Clinical trial is required 7 to 8 years in one, and then only you are able to go for the rest of the thing. Okay. But now, see, in biotech 1.0, speed and scale are years to decades. Sometime it will require the 5 years, 7 years, and sometime it requires the 15 to 16 years. That is the years to decade. The time is requirement, and low throughput. Because of the problem, we are utilizing the live cells. The throughput is always slower. Okay, but in biotechnology 2.0, that we are calling as a synthetic era. Okay, here the things are moving from the weeks to days to weeks instead of the years to decade. Now we are reducing the total time. If you remember the vaccine of the corona, it is came only into the 6 to 8 months of the disease. Okay, yeah, definitely the full clinical trial is not done, but we are able to make a vaccine within the 8 months. It is also a kind of a thing which is we have not achieved previously. Okay, so at least we are able to create, and then we can do the clinical trial, which is the requirement of the process to get something fruitful. Okay. So you can assume what is the things the AI and machine learning is going to change into the biological sector. Now see one of the very basic example and one of the very most important example also. In year 2001, the scientists, the group of the scientists and the group of the countries, they have met in Geneva and they have decided that we are going to do the human genome project. Means each gene of the human body, what it is performing, what kind of a chemical or what kind of an enzyme or what kind of a protein it is producing in our body. We are going to analyze it by using the human genome project. Okay, and the cost is around $100 billion. You can assume the $100 billion is the cost, and the project is floated into the 2001 only. Just after the 7 years, after the 7 to 6 years, next generation sequence coming in a big way. In 2007, the NGS is the next generation sequencing, where we are utilizing the machine learning and AI for the finding the function of the genomes in function of the genes into the large genomes. Okay. Now, in 2007, the cost of the genome sequencing is reduced to only $10 million. You can assume the 6 years before we are having the 100 billions of the cost of the project, but now we are having only the $10 million in 2027. Afterwards, a new era coming, and the Illumina HiSec X. The instrument name is Illumina. Illumina is the company who has designed this DNA sequence. Okay, HiSec X. Now, in 2014, the Illumina HiSec X is coming, and it will reduce the overall cost of the genome mapping to only the $1,000. So you can assume 10 billion, $100 billion, then the $10 million, then we are having only the $1,000 in 2014. Afterwards, in 2026, the modern benchtop, which are utilizing the AI and machine learning in a big way, okay, they require only $100 to process your whole genome, every gene in your body. Okay, so they require only the $100. So this is the cost cutting, which is happening due to the application of the computational power, the AI, the machine learning, and the programming into the biology. So this is the requirement of the era. See, only the innovation is not important. If we are going to do some innovation and it is very, very costly, then again a kind of a problem is going to be started. What kind of a problem? The countries, the persons who are not able to of the money, they are not lack of the humanity, basically, for the persons who are able to achieve, able to pay the money. Okay, again a kind of a thing which we are required to look into by ourselves. Now, if we are talking about the digital synthesis and modeling, what do you mean by the digital synthesis? Digital synthesis means, first of all, you are required to create something by using the software. Okay, you are required to do every research, like the molecular modeling, like the generation of the new drug molecule, like you are going to check the drug molecule with the molecular modeling inside the computer only, where we are mostly utilizing the computer-aided drug design software. Okay, computer-aided drug design software, because if we are directly moving toward the lab and if our results are not there, the loss is very large. Okay, so before going to the lab work, we are required to proceed every model into the laptop, and it is digital synthesis and the modeling, through which you are going to reduce the overall time required as well as the total required to proceed further. Print and translation. Once you have done the digital synthesis, the digital sequence to synthesize. Okay, these digital sequences mostly in the form of the ATGC only. These are the DNA gene code. Okay, These gene code are basically responsible for what? Forming the new kind of a protein and new kind of a character. It is going to provide a new kind of a character to the individual. Now, synthetic DNA code. First of all, we are, according to our choice, we are required to design the synthetic DNA. Through the synthetic DNA code, you are required to modify the transcriptomics. I think everybody know about the transcription. Transcription is that is we are going the DNA is going to transcribe into the mRNA, the RNA molecule basically. That is called as the transcription. Now, modified transcriptomics, according to our synthetic DNA, the requirements are going to change. New kind of RNA polymerase, DNA polymerase now you require. So we are required to modify the transcriptomics according to our new synthetic DNA molecule. Xenobiology protein. Now we are going to do the translation. I think everybody know about how the RNA is going to transfer into the protein, and it is called as the translation. Now the regulated translation is required. Cellular translation machinery reprogrammed to incorporate the non-standard amino acid. Because you have created the synthetic DNA, it is going to create an amino acid which may be new for the organism and building the material which is unavailable in the natural world. Okay, some material which is not the available naturally in your earth. Okay, now 3.2 billion base pair. I think everybody know that the human genome is having the 3.2 billion base pairs. Now, synthesizing the whole genome. Biotech 2.0 marked the transition from editing the single gene. Abhi tak we are targeting only the single gene modification by using the mutation, by using some genetic engineering tools and techniques. Now we are writing the massive synthetic chromosome. Okay, new chromosome. We are going to design new chromosome. Whole genome design is currently shifting from viral and bacterial target toward the complex eukaryotic organism. Till now, we have applied this, but only to the microorganism because their genome is less in size. Okay, and we are having few of the things with us also. So, but now we are targeting towards the major organisms, humans, eukaryotic organism. Okay, we are creating the new synthetic whole genomes by editing at the mega base scale. Researchers are constructing the yeast strain, one of the yeast strain which is utilized into the pharma industry, that is the Pichia pastoris. If you are having the time, you can search about the genetically modified Pichia pastoris. It is in between the eukaryote and prokaryotes. Okay, it is having the assembly of the protein modification, which is called as the post-translational modification, and now mostly utilized into the pharma industry to produce the protein for the human consumption. And it is a purely synthetic microorganism which is developed by the scientist. Now, if we are talking about the tools of the trade, synthetic core. What is the core? Core is only the base pairs, that is the DNA, that is the gene into the DNA. Okay. Now, precision genome writing. First of all, moving beyond the primary genome Caesars. Caesars, I think you have heard about the few of the things, that is the endonucleases. There are certain enzymes are available which can cut the DNA at internal site or the external site. Okay, but at a particular place only. But now we are converting the individual chemical bases directly without the double-stranded DNA break. That you are not required to change the DNA structure. You are just required to change one of the gene in one of the base pair inside the gene. Okay, it is going to create a new kind of a entity. Prime editing, the search and replace the word processor of the genome. Prime editing means we are going to change the whole gene, one of the gene which is responsible for the creation of the diabetes. The gene in that way that the diabetes is not going to be happen, then it is the part of the biotechnology 2.0. till now we are what we are doing, actually we are preventing the disease. Okay, but now we are going to what, we are modifying the gene, we are modifying the DNA, so that disease is not going to be prevailed into the individual. Okay, now these are the DNA assembly. Yeah, please see here, miniaturization, that is called as the lab on a chip. Okay, I think everybody know about the silicon chip. Without the silicon chip, our life is not possible. Okay, our mobile phones, our laptops, our televisions, everything which is governed by the electricity, they are having the one or more types of the chip in it. Okay, now miniaturization is basically lab on a chip, is a new concept. Okay, that is, here we are going to make a chip by using the DNA. Here we are going to make a chip by using the specific protein. Okay, advanced chip can integrate the human cellular system onto the microengineered matrices. See, there are always a problem with the computing power. That is, the computing power is solely dependent onto the chip. But your silicon chip is also having their own capability. You cannot create the chip more than the 2 nanometer, or maybe in future 1 nanometer. Okay, but it is not possible. So how we are going to increase our computing power? There are the one of the way which is called as the utilizing the DNA as a material onto the chip for the storing of the information. Okay, that is basically called as your micro-engineered matrices or micro-engineered chips. Okay, these recreate complex organ dynamics. Like chip on a lab on a chip means you can have the kind of a organ, the miniature organ, so that you can simulate the concept. You are having the heart beating chip, you are having the lung breathing on chip onto the silicon. Okay, enabling rapid and non-invasive pharmacokinetics. By using this, you can check what is happening if you are going to eat the paracetamol, how it is going to excreted from your body, what is the side effect of the paracetamol onto your body, without the actual human. Because see, till now, when we are doing the when we are doing the clinical trials, there are the so many cases are there where we are having the problem of the side effect. Okay, so by using the organ onto the chip simulation softwares and these kind of a chips, we are going to remove the human individual for the clinical trials. Yeah, at the last stage, we definitely use the humans, but in a very low number. Okay, at this time, you are required to have around 10,000 individual to check your one of the drug. So the side effect is going to affect all the 10,000 individuals. Okay. Now, robotics and automation. Robotics and automation is basically going to work into the healthcare sector in a big way. You can see after the 10 year or maybe the 8 to 10 years, we are going to see some of the robots which are going to have the most of the doctors. Okay, because you have heard about so many problems when the individual or the humans are involved into the operation. The chances of the contamination are always there. The chances of the manual error are always there. By using the robotic automation for the surgery, by using the robotic automation for the experimentation, we are going to reduce the error, the human error or the errors which we are creating into the scientific experiment. This is going to increase our overall time of research into the 10,000 times. Okay. Now, computational biology and AI. If we are talking about the computational biology and AI, the whole biotechnology is now shifting towards the computational biology and AI. We are not telling that the lab work is going to end. Lab work is always there. Okay, but these computational biology and AI is going to reduce your hectic work, which is required the human intervention. By using some of the software, by using the robotic automation, by using the AI tools, we are now able to develop some of the basic mechanisms, okay, by which we can perform our experiment without the errors. Okay, neural networks. I think everybody know about the neural networks in your body, how the neurons work, how the axons are play a role into the your signal transfer. Okay, neural networks are basically the kind of a thing which are related to your brain. Okay, now we are talking about the artificial neural networks. Those are called as the ANN. ANN is part of your AI machinery. Okay, deploying the deep learning or architectures to decode the complex organic biological problems. Okay, this is called as the application of the artificial neural networks. Now, protein engines. What are the protein engines? If you have not heard, please visit the AlphaFold. AlphaFold is a kind of a tool which is provided by the Google DeepMind. Google is having their subsidiary DeepMind, which are basically working onto the advanced AI and machine learning. AlphaFold is a kind of a software which will provide, you are just required to provide the 2D structure of the protein. 2D means just the base pairs. Amino acid, what kind of amino acid the protein is having. And the AlphaFold is going to create everything for the protein structure prediction. Use generative AI models to draft the novel de novo molecular structures. You can just tell them what kind of protein you require and what function you require from that protein. And by using the de novo molecular modeling, they are going to create a new kind of a protein. Okay, and they predict their 3D folding and how it is going to work also. Cellular modeling, simulate the entire metabolic pathway. Just take an example. Till now, to produce something biological component, maybe the enzymes, maybe the antibodies, maybe the antibiotics. Okay, we are dependent onto the microorganism. We are dependent onto the yeast. Okay, but in future, by utilizing the cellular modeling, we can simulate the entire metabolic pathways of the eukaryotic cell also, and organ scale biological dynamics to evaluate the drug candidate in silico. Now you are not required to directly go into the field and check the drug efficacy onto the humans. You can check the drug efficacy onto the chip also by using the cellular modeling. And by using this, if everything is fine, your constructed protein do not have any side effect onto the humans, then you can go for the human trial. Okay. Otherwise, first you can do everything, modeling, simulation, creation, everything in a computational biology only. Okay. Now, DeepMind AlphaFold, that I have told you, one of the biggest thing the biology is having. And if you know about it, the scientists which are the creation of the behind the creation of the DeepMind AlphaFold is winning the Nobel Prize of 2025 or 2025 also. Okay, cracking the protein folding challenge. First of all, you are just required to protein the 1D amino acid. 1D amino acid means only the structure: methionine, lysine, glycine, cysteine, these kind of a structure. A simple chain you are required to provide. It is also called as the polypeptide chain. Okay, by using the polypeptide chain only, a 3D structure is going to create it. Okay, now you can assume just 10 years before, 3 persons do their PhD onto the one protein only. For the structure prediction of the protein will require 15 to 20 years. And one of the classical example is one of the structure prediction of the protein require 50 years also. You can assume the protein function, the protein structure, only the protein function and structure require the 50 years. Then how the rest of the research we are going to do? These are the major bottleneck into the research and into the biotechnology also. We can do something today and we are going to have the result after the 10 years. Okay, that is a very large amount of time. Now, deep learning models have solved this problem. predicting the protein shapes down to individual atoms within minutes. Minutes means your AlphaFold require around 20 to 30 minutes only. In 20 to 30 minutes, this algorithm is going to do the work which we are able to perform into the 15 to 20 years. Okay, so you can assume that everything is going to be speed up, and if everything is going to be speed up, the innovation scenario is also going to be speed up. Okay, global database of the life's machinery. If we are calling about the global database, means millions of structure, gene pool, the natural gene pool. How many organisms we are having onto the earth? Microorganism, eukaryotes, prokaryotes, everything. Okay. Then the genes of these organisms are called as the natural gene pools. Okay. Millions of structure modeled across the animal, plants, and microbes. Now, what the scientists are doing? The scientists are creating a kind of a machinery so that we can assess if anybody is going to have a disease after the 2 years. The individual must know it that after the 2 years, because your DNA is modified in that way, it may be possible you are going to have the cancer after 2 years. Okay, so this is the scenario. This is the application of the AI and machine learning in the case of the biotechnology 2.0. Now, de novo protein generation. De novo means new protein, which is not naturally available onto the earth. Okay, so new protein synthesis means generative bio AI. First of all, using the diffusion models, we are going to create a new generative bio AI models. Okay, generative AI in the chat GPT. Okay, the bio AI generative GPT. Okay, what is the role required to provide it the sequence, your essential sequence? Okay, it is going to create the new sequence. It is going to predict the new sequence. Engineered capsids, algorithm designed to customize the viral envelopes capable of slipping. Why we require to engineer the capsid? Basically, capsids are the part of the viral genome which do not have the gene which is going to create the disease. By using the engineered capsid, we are able to make the new vaccines, new kind of a vaccines for the various kind of a disease. Okay, now, and which may be utilized for the various kind of drug delivery system also, gene therapies also, smart biosensors. Like the biosensors are available. If somebody is wearing the smartwatch, they are checking your heart rate, they are checking your number of the steps, they are checking everything. Okay, but now we are moving towards the smart biosensor. Smart biosensor means they are highly specific towards the one kind of a thing, creating switch-like protein so that structure and molecular signal can be detected into the disease biomarker. You are just required to utilize that sensor, and that sensor is going to provide you what kind of disease you are having by analysis of your sweat or by analysis of your blood only. You are not required to provide the blood also. You are just going to put the sensor onto your skin, and through the skin pores and by using your sweat, it will provide you the food is going to be consumed in your body, and it is going to produce the energy and and it is also going to create the building blocks, the protein, the amino acid, everything which are required for the proper function of the body. Okay, so in this case, computational metabolic modeling will be utilized in a big way to remodel the metabolic pathways so that produce the desired chemical large number. There are the to the metabolic pathway cells are highly specific. They are going to produce some chemical only as the requirement of the cell, after which their machinery is going to shut off. Okay, to continue this kind of a production, what we require? We require to modify the specific gene which are responsible for the metabolic pathway. If we modify the specific gene and our metabolic path to be shut off, then they are the same kind of a chemical. It is going to create a new opportunities into the pharma sector. Same kind of a chemical, same kind of a insulin, same kind of a interleukin, same kind of interferon. Okay, so these are the few things. What are the application of the AI and machine learning into the field of the biotechnology? Now, why we are calling it as a biotechnology 2.0? Only one thing is there. Now we are moving from the solely lab work towards our computational bench tops. Okay. First of all, every research will be done by using the computational benchtop. Then once we are going to be so sure that our models definitely going to work, then only we are moving towards the lab. It will reduce our overall spend of the money into the research and innovation, as well as it will improve our chances of success instead of the error. Okay. Now at the last, I'm going to tell you about the precision medicine and the therapeutics. I think you have heard about the precision medicine. If you have not heard, please see. Precision medicine is an individual. Okay, precision medicine means every individual is having their own genome. Then the drug, some of the drug is having the more side effect in a particular person, but the same drug do not have the much of the side effect into the different person. Okay, this is because our genome is different. Our metabolic pathways are working slightly different. We are not talking about we are entirely different, but our metabolic pathways are working slightly different. Okay, so sometimes the same drug which is not creating the side effect into the other individuals, it is going to create a side effect into the one of the individuals. Okay, so precision medicine is a kind of a technology which is the coming in a big way into the future. Precision medicine means we are going to provide the drugs according to the genome of the individual. If you are having the fever, everybody is not required to utilize the paracetamol only. Or if everybody is required to utilize the paracetamol, it is not necessary to everybody utilize the 650 milligram of the paracetamol only. It may be possible for someone 200 milligram of the paracetamol is sufficient. Then it is not required to provide the 650, because the higher doses of the medicine always going to create the side effect. Okay, so this is going to be highly specific, highly specific. The individual, according to the genetic makeup of the individual, the drugs will be provided accordingly. And now the drugs are going to be manufactured and made into the same lab, so that the time once your doctor is seeing your results, then what is going to be happen? The medicines are also created there and then only. Okay, so this is the precision medicine according to the individual genetic makeup. We are going to follow it. Now we are ending this session here only, and yeah, Dalvinder ma'am, you can start further.
Yes, thank you so much to our today's expert, Dr. Himanshu. It was a wonderful session. Can we take the questions now?
Definitely, ma'am.
Yes.
Definitely.
Thanks for giving me chance to address your questions now. Let's take the Gaurav question first. Gaurav is asking what career opportunities are emerging in the field of biotechnology 2.0 for students and researchers.
Biotechnology 2.0, as I have told you, it is the biology as well as the computer, artificial intelligence, the machine learning, everything is going to be amalgamated here. Okay, the new kind of a job that is the NVIDIA, Microsoft, Google, everybody is investing billions of the field of your synthetic biology. Okay, synthetic biology is going to create the so many jobs into the future. One is the synthetic biologist, second one is the data analyst, third one is the big data analyst, and fourth one is the your smart bio manufacturer. Okay, these are the few fields which are not existing till now and now going to be created in a big way. Yeah, I think I
Thank
have
you so much.
answered the question. Yeah.
I hope you got your answer from our expert. So, Arpan, Devansh Kumar Pandey, Minal, if you have any question, you just write in the chat box, so sir will definitely answer you. So, Pariniti is with us. Pariniti is asking, how can artificial intelligence and automation accelerate advancements in biotechnology?
Artificial intelligence, if you know about the how the artificial intelligence currently working, it is a kind of a model which we are required to train first of all. Then we are required to utilize that model. If you utilizing the chat GPT, because most of the person are utilizing the chat GPT nowadays, okay. Initially, chat GPT is providing the random answers. But once you are talking and you are providing about your wish or what you want from the chat GPT, the answers are going to change. It is called as the self-learning module of the AI. Okay, the self-learning module, how we are going to utilize the self-learning module in biology. Now we are developing the so many algorithm for the analysis of the whole genome, for the analysis of the DNA, for the analysis of the different kind of a chemical which are produced inside our body. Okay, so the AI is going to reduce the work in a larger way. When we require initially 5 years before, just 5 years before, we require 10 to 15 years for the development of the new drug. That is reduced to 5 to 6 years nowadays. Okay, and in future it may be possible that it will reduce to 1 to 2 years only. So the biotechnology 2.0 is going to help in the healthcare sector, in agriculture, in food biotechnology sector also.
Thank you so much. I hope, Parinidhu, you got your answer. So, Vinal is asking, sir, I have wondered if the smartwatch would also come with a dose of protein, like heartbeat and other shows in smartwatches. Can we show protein in smartwatches?
Can you repeat the question?
Yeah. Sir, I have wondered if the smartwatch would also come with a dose of protein,
Okay.
like heartbeat and other shows in smartwatches. Can we show protein in smartwatch?
Definitely. See, there are the possibilities are there. Till now, we are not able to make any wearable device which can check the sweat for the protein detection. Okay, but research are going into the same direction. In future, we are going to be smart humans. Okay, in future, everybody is going to convert into the smart humans, and continuously our body is checking for the changes. Changes means how the DNA is modifying, how the proteins are making in our body. So in future, it is definitely we are going to have that kind of wearable also. Till now, we are having the sensors in smartwatch which can check the blood pressure, which can check the heart rate, and which can check the your number of steps you have walked in a day. Okay, so just wait. It is just a time when we are going to have that kind of a wearable also. Definitely.
Thank you so much. I hope, Minal, you got your answer from our expert. So if anyone have any question regarding the admission, they just write on us at admissions@lpu.co.in and you will do WhatsApp chat with us. Our WhatsApp number is 985256900. I'm repeating the number. It's 985256900. And once again, thanks to our expert, Dr. Edu-Webinar. And if anyone wants to access this webinar again, they can visit our website, that is lpu.in under admissions, then LPU Edu-Webinars. You will find the past and the upcoming webinars. And you can reassess this video, this webinar later on YouTube as well. Stay connected with us for future webinars. Until we meet again, with the permission of my series, I will be signing off this webinar. Take care and goodbye. Have a beautiful day.
For providing the